Okay, so we are looking at the classes of amino acids. So we find that these amino acids are actually going to affect the shape of a protein depending on the chemical nature of its side group. So each amino acid will affect the shape of a protein depending on the chemical structure of its other group of the side group. So for example, you find that certain portions of a protein strain or a protein chain with numerous non-polar amino acids. So these tend to fold into the interior of the protein by hydrophobic exclusion. So since these contain non-polar bonds, in other words, since they are non-polar, so they're going to fold inwards into the interior because they are hydrophobic and not hydrophilic, so away from the water. And then the other point we need to note is that functional groups are going to affect the reactivity of each amino acid. So we find that there are several side chains that are found in amino acid. And those side chains may contain functional groups, different functional groups. So remember, each amino acid is going to contain an amino functional group and a carboxyl functional group. In addition to that, these side chains would contain the sulfhydryl group and the hydroxyl functional groups. So these are going to affect the way an amino acid is going to function and react as well. So I find that under special circumstances or under right conditions, these functional groups are able to participate in chemical reactions. So for example, you may have, when we talk about the sulfhydryl functional group or the SH functional group, if it is in the side chains, we find that it can form disulfide bonds. So these help to link different parts of large proteins. So these functional groups participate in chemical reactions. And then we find that in contrast, some amino acids only contain chains that are devoid of functional groups. So they may just consist of hydrocarbons, just carbon and hydrogen atoms. So these will rarely participate in reactions. So I find that these amino acids will influence the function of the proteins depending primarily on their size and shape rather than their reactivity. Since they rarely react, so they're going to affect the function of the protein depending on their shape and size instead of their reactivity. And then also of the polarity of the side chains, these are also going to affect their solubility, the solubility of the amino acid. So the nature of the side chain will affect the polarity and henceforth is going to affect the solubility of an amino acid in water. So for example, non-polar chains, these they lack charged or high electronegative atoms, which are capable of forming hydrogen bonds with water. Since they lack these high electronegative atoms and they also lack rather charged atoms, these tend to be hydrophobic. So it means that these are not going to be able to interact with water. So instead of them dissolving, they are going to coalesce in aqueous solution. So the side chain in this case has affected the solubility of an amino acid. However, when we look at the polar or charged side chains, these are going to react or interact readily with water, and henceforth these are hydrophilic. So these hydrophilic side chains can easily dissolve in water. So, as we mentioned earlier, these side chains hence are going to also distinguish the different amino acids and group them to either be acidic, basic, non-polar, or uncharged polar. So we have the non-polar side chains. These amino acids contain non-polar side chains. So when we look at these, these, we find that the non-polar side chains, they are not going to contain any charged or any electronegative atom to form any hydrogen bonds. Okay. So the non-polar side chains, they contain no charged or any electronegative atoms to be able to form hydrogen bonds. So these are examples of those amino acids with non-polar side chains. Then we have the polar side chains. So these are going to have partial charges. So these are able to form hydrogen bonds. So you have polar charges. So partially positive, partially negative. So charges. Then we have the electrically charged side chains. So these are able to form ionic and hydrogen bonds. So the side chains are given a different colors we can see. So these are able to form ionic and hydrogen bonds. How do we determine that an amino acid from the table that we've seen is acidic or basic or non-polar? So you look at the side chain. You look at the side chain. So you look at the side chain. Does that side chain have a negative charge? If it has a negative charge, it means that it has lost a proton, so it must be acidic. Easy. So you look at the negative charge. Has it lost a proton? Then it is acidic. Then does that side chain have a positive charge? Then it means that it has received or taken a proton. Then it means that that amino acid should be basic. And then if the side chain is uncharged, you look at the side chain, does it have an oxygen atom? Does it have an oxygen atom? If so, the highly electronegative oxygen will result in a polar covalent bond. And thus, that amino acid is then grouped as being uncharged polar. So you check, is there a negative charge? If yes, it means that it has lost a proton. And if it has lost a proton, it means that it is acidic. If it has a positive charge, it means it has taken a proton or a hydrogen. Then it means that it is basic. Then if it is uncharged, you ask if it has, you look if it has an oxygen atom. If that is it, it means that that oxygen atom is going to form that polar covalent bond. And hence for that amino acid will be uncharged. And then if the answers to all the above is no, that it means that you are looking at a non-polar amino acid. I hope that is clear. Okay, just a minute. Okay. So we are saying, you look at the charge. Is it positive or is it negative? Is it negative or is it positive? And then if it is uncharged, it does not have a charge, you see if it has an oxygen atom. It has an oxygen atom. It means that that high electronegative oxygen will result in a polar covalent bond. And then you term that as an uncharged polar amino acid. And then if the answers to all the above questions is a no, then it means that you are looking at a non-polar amino acid. You're looking at a non-polar amino acid. So for example, on the polar side chain, you look at the molecule. So remember, this is a polar amino acid. So when you, just a minute, when you look at this amino acid, you look at the chain, you look at the charge. Is it a negative charge or is it a positive charge? Is it a negative charge or is it a positive charge? So for example, in aspartate or glutamate, you can see there's a negative charge at the side chain. Aspartate has a negative charge on the side chain. So it means that this amino acid has lost a proton. So it is acidic. However, when you look at a lysine or arginine and histidine, you look at the side chain, you are going to see the positive. So it has a positive charge. It means it has taken a hydrogen or a proton, in this case, a hydrogen. So this means that the amino acid is a basic amino acid. So just look at the side chain, we're able to determine whether it's basic or acidic. And then we have what are termed as essential amino acids. So look at these essential amino acids. These are amino acids which animals cannot synthesize in the sufficient amounts to be able to meet the body's needs. So these are obtained from our diet. So they are essential, they are termed essential because these have to be obtained from our diet. We are unable to synthesize them in the right amount. We are unable to synthesize them in the right amount. However, when you look at plants, plants are able to actually make all the 20 amino acids. All animals are unable to synthesize some of the amino acids. So leading to having this term of essential amino acids. So some of the examples of essential amino acids that we have for humans, we have isoleucine, we have leucine, We have lysine, methionine, phenylalanine, threonine, tryptophan, valine, and histidine. Or in children, also arginine is not produced in sufficient amount to produce or to support growth. So usually it's also added in case of children, arginine. So these are the essential amino acids. We are unable to synthesize them in the right amount. So no wonder they are termed as essential amino acids. Now let's look at the peptide bond. The peptide bond. So these are the bonds that we find